So I will talk about the future of structural biology. This talk is viewed through the lens of the pandemic, and by definition, then it is global. I'll touch on the protein data bank and then say something about quantitative about the role of structural biologists and the PDB on drug approvals. I hope you take this information away and use it as ammunition. There are 2 published papers now. And then I'll actually highlight some of the cutting edge contributions that structural biologists have made during the pandemic. Touch briefly on integrative structure determination, which I believe to be the future of structural biology. And I'll leave you with a sobering postscript that makes, I think, an important point about preparedness for the next time a coronavirus jumps the species barrier, because there will be a next time. We just don't know when. So the Protein Data Bank is celebrating its 50th anniversary of continuous operation on the 20th of October 2021. We're now in our 50th year. This was the first open access digital data resource in all of biology, founded in 1971. And it's grown to an open access archive of more than 170,000 structures that since 2003 has been managed by a global consortium that includes the US, Europe, Asia, and a specialist resource, Biomag ResBank. The work that we've done recently looking at impact on drug approvals by structural biologists and by the PDB is summarized here. There were 210 new drugs approved by the US FDA between the beginning of 2010 and the end of 2016. And approximately 6,000 PDB structures and open access to these structures facilitated 184 of these 210 drug approvals. The impact ranged from understanding target biology all the way to structure-based drug discovery, and I'm specifically highlighting here on the right the discovery of vemurafenib for the treatment of late-stage melanoma, targeting a mutant form of the BRAF protein kinase that is abnormal, that's mutated in the same way in 50% of patients with metastatic melanoma. There are 2 papers from our team that are provided here at the bottom of the slide. The Westbrook and Burley structure, and then the Westbrook et al. Drug discovery today that was on the cover of the May issue. Those papers actually provide hard numbers that show the importance of structure and structural biologists in drug discovery. The second paper documents that more than 70% of the new anti-cancer drugs that were approved 2010 through 2018 were the product of structure-based drug discovery. That's more than 70% of 54 new anti-cancer agents. If nothing else, that should be a significant statistic for funders and others thinking about the importance of structural biology. So, Joel talked briefly about icing on the cake or silver lining in a cloud. I'll say that it's an ill wind that doesn't blow somebody good. And the fact is that the COVID-19 pandemic has created enormous opportunities for feedback by structural biologists around the world. So you know the timeline. Influenza-like illness in late 2019, causative agent discovered and the genome sequence released in January of 2020. The first PDB structure of a SARS-CoV-2 protein was released by the PDB in early 2020. It came from Ziho Rao's group in Shanghai. It was the main protease, or NSP5. Today, there are 1,030 coronavirus structures in the Protein Data Bank. 550 of those are SARS-CoV-2 structures. The remainder come from other coronaviruses. They are allowing us to see the unseen enemy, which is depicted here on the right. This is a SARS-CoV-2 virus inside an aerosol droplet. This is mucin and antibodies and things like that. And the transmissive agent of this illness is this droplet, featured in a watercolor painted by our molecular artist, David Goodsell, who's a faculty member at Scripps and also at Rutgers. We've put a lot of effort into prioritizing the management of the depositions of COVID-19 related structures. We've encouraged all of our depositors to release immediately, and virtually all of them have. We're providing consistent biocuration and a great deal of information about COVID-19 resources that you can find on our webpage, rcsb.org. And for the public, we've produced videos of the type that you see here, explaining the importance of hand washing in terms of demolishing the virus. As we look across the SARS-CoV-2 proteome, which includes 30 proteins, we have structural information either directly from the protein data bank or indirectly through computational modeling for 29 of these 30 proteins. I'm going to talk about in a little bit of detail the main protease that's responsible for cutting up the polyprotein. That together with the papain-like protease is what processes this very long polyprotein into 16 different segments. I'll touch on the RNA-dependent RNA polymerase, the helicase, and then I'll also talk about the Eprotein and the nucleocapsid protein, all of which have been studied by structural biologists in some way. So starting with macromolecular crystallography, the mainstay of structural biology, notwithstanding the importance of cryo-EM. The big news here is fragment screening by crystallography. Much of it has been done at Diamond, as you know, with the PANDA structures. Today, there are 200 PDB structures of the NSP5 main protease in the protein data bank. 116 of them are PANDA structures of small molecule fragments bound to the target that have been detected by crystallographic screening. These 116 PANDA structures plus another almost 50 co-crystal structures with various inhibitors are informing discovery of NSP5 inhibitors. And shown here on the right, I'm featuring one of the structures that came out of the PANDA series for NSP5, showing this small fragment bound non-covalently in the active site. The two key residues in the active site are the histidine here and the cysteine here that are responsible for catalysis, for cutting up the polyprotein into its constituent pieces. 3D-EM has had an enormous impact on our understanding of SARS-CoV-2 proteins, and I'm focusing here on the work that's been done on the RNA-dependent RNA polymerase, which is a heterotetramer consisting of the non-structural proteins 7, 2 copies of NSP8 and NSP12. This is responsible for duplicating the viral genome and making subgenomic RNAs. It's the target of remdesivir, the only approved drug in the United States for direct treatment of SARS-CoV-2 infections. There are 15 SARS-CoV-2 structures of this complex in the PDB and another 2 from SARS-CoV-1. These structures are informing discovery of second-generation RNA-dependent RNA polymerase inhibitors. The goal would be to improve the potency and selectivity. Remdesivir is not a very good drug by the standards of modern drug discovery. It's a repurposed Ebola agent. And a key change to the pharmacology would be to make an orally bioavailable inhibitor of RDRP. It's shown here on the right. The big subunit NSP12 is catalytic. Remdesivir binds here. NSP7 is here. And then there are 2 copies of NSP8 are responsible for stabilizing the duplex RNA as it's produced by the enzyme and preventing its dissociation. I tell the students at Rutgers as they're coming into our program in quantitative biomedicine, the graduate students, that tomography is the structural science in terms of measurement of the future. And I'm featuring here work from Klein et al. That was reported recently in Nature Communications, showing the power of tomography to visualize SARS-CoV-2 in the cell and to actually understand some of the details of virion assembly in the endoplasmic reticulum Golgi complex. And you're seeing one such virus being assembled here in the ER Golgi complex. NMR remains an important technique, notwithstanding the fact that the number of new structures coming from NMR deposited into the PDB is declining on an annual basis. Here's an important structure of the SARS-CoV-2 nucleocapsid protein, or N protein, the N-terminal domain of which binds to RNA, and there's a recently published NMR structure of the complex between a short duplex RNA and the nucleocapsid. In total for the nucleocapsid, there's 24 different structures that are informing our understanding of the fundamental virology of the system. The combination of the N and C terminal domains both bind the RNA and provides the organization of the genome to compact it within the nucleocapsid, which is a confined space. Important to know that the genome of coronaviruses is the longest of any of the known RNA viruses, single-stranded RNA viruses. The Eprotein is another drug discovery target. It's a pentameric ion channel, a viroporin, if you will. It can be found in the endoplasmic reticulum Golgi intermediate complex, the ERGIC, and also in the virion bilayer. Within the virion bilayer, it ensures osmotic stability of the capsule and also is responsible for bending the lipid bilayer. And this is probably what allows what ensures that the lipid actually forms a compact envelope around the nucleocapsid. Also embedded in the lipid bilayer, of course, are the spike protein, and there's been an enormous amount of cryo-EM work on that, particularly with monoclonal antibodies neutralizing that are bound to the spike. There's also another integral membrane protein in the bilayer known as the M protein. There's a relative paucity of structural information for the E protein, just two structures, one for SARS-CoV-1 and one for SARS-CoV-2. Importantly, this new structure on SARS-CoV-2 is a solid-state NMR structure, which provides the first high-resolution look at the pore, which you're seeing here. It's these 5 central helices that actually determine the ion selectivity and the passage through the membrane. So solid-state NMR I also view as being very important. Now, as people are trying to understand more about the virus, they're tackling larger and larger complexes, and I want to introduce here the NSP13 RNA helicase. This is essential for unwinding the RNA secondary structure in the viral genome. The 5' segment of the RNA genome has a very extensive secondary structure that intrudes on the coding region. It's known to work in concert with the RNA-dependent RNA polymerase. As of today, there are 55 SARS-CoV-2 NSP13 structures in the protein data bank. 51 of these come from the PANDA effort at Diamond, probing the binding properties of the enzyme, and specifically how small molecules will fit here in the active site of the helicase. These represent guideposts for starting points for discovery of helicase inhibitors as antiviral agents. I want to move now from this structure of the RNA helicase, just the individual protein, to this recently published cryo-EM structure that shows how the helicase interoperates with the RNA-dependent RNA polymerase. So on the EM grid, you have the RDRP, NSP7, 2 copies of 8, a copy of 12, the catalytic subunit, and 2 copies of the RNA helicase. They bind in this fashion. I'm going to show you another view which is more informative. This is the back side of the view that I showed you earlier. If we now look directly down onto the active site from above, and we can see the duplex RNA here in the active site, this is where remdesivir would bind. The 2 copies of the helicase extend out from the mouth of the active site, where they would be able to unwind the RNA as it's about to enter the active site to ensure that the enzyme remains processive and doesn't get stalled on the genome by the need to actually wait for the secondary structure to open up. This brings me to integrative structure determination, which, as I said, I believe is the future of structural biology. As our appetite grows for understanding larger and larger systems, cryo-EM will not be enough. X-ray crystallography will not be enough, and it will require the integration of measurements from a variety of different complementary methods, some of which are listed here, and the results of computational modeling of one kind or another, and chemical cross-linking to establish a set of spatial restraints. And then using a satisfaction of spatial restraints calculation, one can go from a collection of individual structures to an ensemble of optimized structures. The precision of these structures is probably on the order, at best, on the order of a few angstroms, not a few tenths of an angstroms, which is the precision of macromolecular crystallography. But it's enough in situations like this with in terms of studies with the nuclear pore complex to understand the architecture of the system and begin to develop hypotheses that can be used in functional tests of the system to understand how a machine as complicated as the nuclear pore complex works to allow selective passage with carrier proteins of cargo both in and out of the nucleus. There is a place to archive these PDB Dev, these integrative structures. It's called PDB Dev. This is a prototype archiving system which is currently funded at a low level by the National Science Foundation as the methods for both archiving and for validating the integrative structures are developed. Our long-term goal is to transition both the archiving system and all of the structures into the protein data bank, so there'll be a one-stop shop for deposition of both the primary data and the integrative structure. And these integrative structures, some of them will have all atom representations, such as some of the structures shown here. Others will have multi-scale representations, and in some cases it will be a mix of atomic and multi-scale representations. All of these are going to be possible as we transition these integrative structures from PDB Dev into the PDB archive. So with that, I'm going to close with a sobering postscript that underscores the importance of pandemic preparedness and of us scientists making the case for opportunities that should not be squandered when they present themselves. So I'll return to the main protease, NSP5. NSP5 sequences are very highly conserved across all coronaviruses. Between SARS-CoV-1 and SARS-CoV-2, the sequence identity is 95%. And of course, the structures, the 3D structures of these main proteases are extremely similar across the coronaviruses. The RMSD for SARS-CoV-2 NSP5 versus that of CoV-1 is less than 1 angstrom. If one considers only the residues, only the non-hydrogen atoms that are lining the active site, there's one here and one over here, it's an obligate homodimer. The RMSD for these non-hydrogen atoms is even smaller. It's less than half an angstrom. Now, the structure I'm depicting here in green in this overlay was determined by SGX Pharmaceuticals, where I was the chief scientific officer back in 2003 at the height of the SARS epidemic. We published we put the data into the PDB immediately and released it. We published the structure, if you will, in the New York Times. That's the only New York Times published structure in the entire archive. And then I went to the heads of major pharmaceutical companies still involved in infectious disease research and asked them if they would partner with us on structure-guided discovery of a SARS-CoV-1 NSP5 inhibitor. And they all politely declined on the grounds that they thought SARS was going to go away. No one in the US had died of SARS. And the epidemic had been contained, and there was no economic rationale for them to do so. And there was no move by governments or NGOs to provide additional incentives. Nearly 10 years later, MERS comes along, and that should have been the wake-up call for us to begin investing in a strategy for pharmacologic intervention when the next coronavirus species, the next coronavirus will jump the species barrier. The MERS epidemic proved to be fairly self-contained, although interestingly, because it has an animal reservoir in camels, people are still getting infected with MERS in North Africa, in the Middle East, and in parts of Asia. There aren't so many people dying now because the medical profession has got much better at taking care of people with MERS, but we have not eradicated that disease. And the sad story in all of this is that had we invested a few $100 million in the discovery of SARS-CoV-1 NSP5 protease inhibitor back in 2003 or in 2013, we would have a safe and effective drug against SARS-CoV-2 today. And we would have averted literally millions of deaths and trillions of dollars of economic losses worldwide. Only one country prepared adequately, in my view, for the pandemic, and they focused on immediate public health issues like personal protective equipment, tracking and testing and tracing, etc. And that was the country of Taiwan, where they limited infections to about 500 people, and the number of deaths was on the order of about 25. They made some investment in protease inhibitors but didn't continue. That's unfortunate. Similar investments were made in mainland China, but were also not continued. It's clear to me now that we as scientists need to say something about pandemic preparedness and the role that science can play along with public health measures to prepare for the next coronavirus that's going to jump the species barrier, because it's not a matter, in my view, of if, it's just simply a matter of when. We were lucky the first 2 times, we were extremely unlucky the 3rd time, and there's no reason to believe that we'll be lucky the 4th, 5th, or 6th, or however many times these events occur, as people and animals that carry these coronaviruses end up living closer to one another. So with that, I will thank you for your attention. I'm able to give this presentation because I am backed up by an enormously talented team of people working at the RCSB Protein Data Bank. They're all depicted here. I want to draw particular attention to Andre Sali, who's leading our efforts on integrative structure validation. He's at UCSF. And I also want to recognize the achievements of Professor Helen Berman, who's now an emeritus Rutgers faculty member living on the West Coast, who was responsible for many of the innovations that have made the PDB as successful as it's been since it was moved to Rutgers about 20 years ago. And I thank all of our funders at NSF, NIH, and the US Department of Energy, our hosts at Rutgers, UC San Diego, and the University of California, San Francisco, and acknowledge the contributions made by our partners in the Worldwide Protein Data Bank. And I thank the Instruct organization for involving me in so many interesting ways in your journey. Thank you.